you been? We haven't seen each other in a while.
Yeah, been good. Been good.
Yeah.
Got the tenure letter earlier this year. So
Congratulations.
yeah, feels good. Feels good.
Good.
And
Good.
yourself, you've transitioned into another era of your professional life, right?
Yeah. The joke around the lab is I'm the postdoc emeritus now. I, I went to 2 days a week. Still I turned the headaches over to DC, and he's doing a
Uh,
real fine job. But
yeah, yeah.
scientifically, I'm still very much engaged, and we still literally talk every day and work on the science, et cetera. So I'm loving it, to be honest with you. Just to be able to get rid of the bullshit, it's
All
just
right.
been great for me.
Well, hey, I'm very appreciative for your time. Getting to know you, and particularly in Colombia, was fun and cool because you're a name that I've read, countless times, learning and going through school and whatnot. So it was cool to get to know you. But yeah, thank you
Oh,
for your time.
well, thanks for making me feel old, Kurt. I appreciate that. I hear that a lot, by the way. Oh, I remember reading your stuff. And you know what? In fairness, I did the same thing
Yeah.
to people, over the years. I was like, yeah, I remember I read your stuff in graduate school. So I get it. I know how it is. So yeah, thank you, though.
Well, you paved the way. You paved the way for a lot of people.
Well, I'd say we did. As you know, I came from a very substantial laboratory and brain trust. Let's just put it that way. So, and I had the advantage of being with Bob for those 30 some years. So, pretty steep learning curve, pretty amazing opportunities, and yeah, you're right. We kind of were able to lay the foundation of this area, especially in terms of amino acids and stuff. But people have to realize that Bob did that before me in terms of glucose
Mm,
metabolism and fatty acid metabolism. I came along when he was foraying into amino acid metabolism and was able to benefit from that whole, like I said, learning environment. But I don't think, maybe because there aren't, Bob's 10 years older than me. So as you can imagine, most of his colleagues have passed at this point. But I think people have to realize, if you get back into the early 80s, long before any of you all were born, he was doing substantial work in terms of glucose kinetics and in stress physiology and exercise and on and on and on. So pretty amazing, pretty amazing.
yeah, I was thinking about that because now some of the stuff that you all sort of pioneered, it's more prevalent. It's not ubiquitous, right? But there's more work being done in it. And so yeah, I was wondering how you specifically sort of got into that because my understanding is that the field of exercise physiology, especially when it gets into some of the like real physiological biological measurements, is relatively new. And previously, exercise science programs, or exercise physiology programs, and to the extent they existed, were in a lot of them were in PE departments or PE programs.
Yeah, and kinesiology, you're right.
Yeah, yeah.
Yeah, you're exactly right. Um, yeah, it's an interesting question. And again, I just look back and see how fortunate I was. Um, I did a postdoc at NASA. I don't know if many people know that. I went to NASA to be a postdoctoral fellow, and I spent, I think, about 3 years in that capacity, actually. And at that time, my mentor at NASA was a gal named Helen Lane, who ran the nutrition division. And she wanted to figure out what the story was with protein turnover in astronauts, number one. And number two, like, how do we mitigate it? Because all they knew is that when astronauts went into space, they became weak and lost muscle mass. So she brought me on as a conduit between NASA and Bob Wolf's lab. And so my work in studying protein turnover was actually in Bob Wolf's lab for the most part. Okay. So to her credit, she was the conduit for the setup. And, Bob, of course, has always been collaborative and congenial and, welcomed the, unknown postdoc into his laboratory. and things took off from there. I had to learn. The learning curve was like this. It was like I mean, it reminded me of my military days of doing mountain climbing, mountaineering. It's because that's kind of what it felt like. It was. I mean, I was totally unprepared for it. It was a totally new way of life. This whole tracer methodology was a whole
Right.
subscience into itself. In fact, I went to Bob's first tracer methodology course, which was held out in uh, where was the Olympics held? Squaw Valley, the 1960 Olympics. He had it on that site on the ski slope.
Oh, wow.
And so, I went to this course probably 7, 8, 9 times over those years. In fact, ended up teaching a lot of it, but learned a lot again in the laboratory. And there were some great, postdocs and fellows in there. It was a great collaborative effort. It had the initial feel of taking a sip of water from a fire hydrant. I kind of felt that way for probably 2 or 3 years, to be honest with you. I still joke to the laboratory that my initial conversations with Bob, when I walked out of the office with him, I would ask myself, like, what the hell did he just say? What was he, what I mean? He was so far above me at that point. I was racing to catch up, both in terms of physiology, metabolism, methodology. I mean, it really was like learning another language. I mean, I know you speak Spanish, for example. I speak Italian. It's literally like learning another language. So yeah, they were very formative years, but they served me quite well in sorting out those foundational issues and the methodology, and it turned out well. And at the end of that postdoctoral fellowship, I kind of had a choice to stay there at NASA, or he offered me a position there, and I took the position there because I just knew that my capability in science was 1,000 times greater in that laboratory, and I was right. So that was the foundational steps of my career, if you will.
Did you come to it from because I know back then the potential career paths or academic paths even might be a little bit different. Did you come from it from a like a sport athlete standpoint? Hey, I'm interested in exercise physiology, nutrition, or were you coming from a physiology and or medicine perspective and just
Yeah,
landed with exercise physiology and nutrition, for example?
it was the latter, I would say. I mean, I was always interested in physiology, human physiology, and adaptations of human physiology and ways to enhance performance. But I recognized fairly early. I mean, I came from I did a PhD at Florida State, and I did like nutrition physiology, exercise physiology. They allowed me to kind of go out and get all that stuff, muscle biology, kind of group it all together, which I really appreciated for my future interests. But I also knew that like exercise itself was quite limiting. We're all exercise guys, but the point is, in terms of funding, scientific career, on and on and on, it's quite limiting. And I was very interested in the clinical medicine, the clinical physiology, et cetera. And I got a very early interest in stress physiology because I don't know if you knew this, but our laboratory was actually in the Shriners Burns Hospital, the Shriners Burns Institute. So all the initial work that I had done was in burn patients. So that was, again, a foundational learning moment of like, because it's hard to find any human physiology that's stressed more than a severe burn.
Right, right.
So from that, we were able to characterize pretty much every bit of that over the years, and from that learn like, okay, what works, what doesn't work, what do we need to do to keep the organism alive, on and on and on. And so I don't remember if I did it when we were in Medellin, but I always show a slide of like what I call the stress scale. And so high stress, low stress, which also coincides with muscle loss. So low stress, low muscle loss, high stress, high muscle loss. Like in the early days of burn care, uh, covering a burn is not that hard of a deal. Okay. That methodology has been known for quite some time. But the patient would die in the early days because of a loss of lean mass, not because of the actual burn itself. Okay. So the key became like, how do we keep the patient from losing lean mass? And to follow that on, you may or may not know this, but in almost every clinical instance, lean mass correlates highly with survival, whether it's cancer or whether it's diabetes. I mean, whatever the pathology is, if you can maintain lean mass, the organism has a much greater chance of survival. And think about it, you probably know the reason for this, because the lean mass is the only storage of the body's amino acids. Like, think about it. It's only protein. There's no, it's not like glucose. We have liver glycogen, muscle glycogen, you don't have that. It's like you either utilize the nitrogen or it's excreted because it's toxic to the body after a certain standpoint. So the muscle serves as a reservoir for stress physiology. And that's why I don't remember which lectures I gave or which ones you heard, but I've always said, if the whole body isn't happy, in other words, if the whole body is stressed, it doesn't matter what you do to the muscle, because the muscle is just a reservoir at that point. It's just providing amino acids so that the more important proteins, the liver proteins, the gut proteins, the brain proteins, the heart, those more important proteins to the organism's survival are predicated on the amino acid precursors held in the skeletal muscle. And so recently we did a lot of work in the military, soldiers and all that stuff, which really demonstrated that quite clearly. We just came out of 20 years of Afghanistan, where soldiers would go out and spend several days at a time in operation. So they ate very little. They had a high op-tempo, high exercise. So we kind of mimicked that they were like 30% caloric deficit and then exercise on top of that. And we kind of looked at, over a series of studies, like how many amino acids did they need to maintain like balance in this thing. And it turned out to be quite a bit. It turned out to be, like 40 some grams of amino acids that when they're hypocaloric and high exercise, that was needed to keep the body happy. Okay, so kind of keep that in mind. And we used a free form amino acids because they were, didn't require digestion, easy to get there, etc. So as you know, like 40 grams of EAAs would be like 90 grams of whey or 100 grams of whey.
Right.
Pretty heavy, substantial load of protein / amino acids. So I think that's what people have to understand. That's why I keep harping. I harp at ISSN all the time because we're so myocentric. You know what I mean? Everything's about the muscle, muscle, muscle. But again, anytime the physiology is stressed, like talking to Jackie and Tony Ricci because they deal with the MMA, they lose weight. When they're losing weight, when they're in caloric deficit and still training, boy, you got your hands full there.
Right.
You got your hands full in terms of maintaining proteostasis, body of protein balance, as I call it. So, all these things add up, and it's just not as simple as targeting the muscle like the supplement world and our field and everybody else tells you. And if everybody's happy and healthy, yeah, we can do all the targeting of muscle we can. But once you throw any one or more stress parameters in there, you really have to take that into account.
All right. No, that quote that you just mentioned, if the body's not happy, it doesn't matter what you do to the muscle. You said that at in Colombia, and I actually use that in my classes to kind of make the point of, if we talk about muscle, a lot of it has to do with performance or exercise, and try to bring in the emphasis of skeletal muscle as it relates to overall health and function and risk of disease. And with that, the importance of protein intake. And a lot of people might think like, oh, protein is just for bodybuilders and athletes and whatnot. But to make that point of like you're mentioning, protein is a vital nutrient for not just muscle, but for every single system, tissue, organ in the body. And if it's not getting what it needs, then the muscle is going to suffer, which that in and of itself is going to have detrimental consequences from a health and say longevity perspective, not to say least about performance.
You're right. And so let me flip the coin on you with that. So again, it's another point that you can use in your class. Again, well before your time, in Ireland, the northern and southern Ireland, and in Belfast. Before you were born, there was a lot of uh, protests with Catholics versus Protestants, and they had a number of like starvation studies, starvation in terms of to protest. And if you look at the starvation studies, somebody was prescient enough to like draw blood throughout the entire starvation period. And blood amino acid levels were maintained in the blood right up until the point they died. So in other words, again, when you ran out of lean
Right.
mass, you
Right.
died. The support of the body organs were dependent on the lean mass being able to provide, and once it could not, they died. So again, it's two sides of the same coin, is what I always say. Uh, so again, it shows you the importance of lean mass in this whole equation.
Right. Yeah, I think that's a very important point. Because our students, some of them might be wanting to go into athletics and sport performance and coaching and whatnot, but a lot of them, they want to go PT route or some sort of allied health route, into medicine, something like that, which that the muscle concern, the amino acid concern is going to be outside of performance and athletics, but still incredibly, incredibly crucial.
Yeah, and over the years, we and others have shown the importance of the additional amino acids in terms of rehabilitation, et cetera. Um, Again, like David DC always says, research is me search. So after the surgery, man, I pounded these amino acids like 24/7, and my recovery was great. My recovery is great. Like, in fact, I did a little boxing last night. 6 weeks after the surgery, I did a little boxing last night. So, I, you know, I'm not
Right,
exactly
Tyson?
Wolverine, but my point is that with the proper rehab and the proper nutrition, I was able to kind of move this timetable along. In fact, I saw my surgeon on Wednesday, and I said, what about rehabilitation? He said, well, you've already done the rehabilitation. I mean, what else are you going to do? I said, I just need the green light. He said, you got the green light, but avoid ABC. Okay, fine, thank you, doc, and moving on. So yeah, that's the way it is. And that's just not what I found, but I found that in studies when we looked at hip and knee arthroplasty and many other people. In fact, Kurt, if you look at that ISSN position statement we put out last
Right.
year on the
Right.
EAAs, to me, the most important data there was one paragraph that Bob and I put in there that showed the benefits of amino acids in all kinds of clinical studies. Okay. So there's actually been more clinical work with EAs because, as you can imagine, that's what
Mm-hmm.
gets funded, right? That's what the
Yeah.
NIH
Yeah.
is going to fund. They're not going to fund exercise work. And, we did like 25 clinical trials in the development of these patents, but there's probably been 75 clinical trials done now with some aspect of EAAs and never a negative finding.
Right.
Think about that. Always a positive finding in terms of, sorry, I got 4 dogs. And that's what you heard. So, but the point is always a positive findings of EAAs and outcomes, certainly clinical outcomes. So, again, not magic, but it just tells you the importance of the substrate, number one, and more importantly, the importance of a specific delivery of that substrate and its effect on physiology. I think that's what people keep missing. It's like we were so conditioned to proteins, which we all, the human being was raised on protein, right? They killed the deer, you ate it, you know what I mean? We were hunter gatherers, and to this day. And then, oh, by the way, whey protein comes along, a great protein source, by the way. We were throwing it away for many years, and then we realized, holy shit, there's really a lot of benefit to whey protein and these other proteins, etc. What I think we've done over those years is, I'd like to say I've been more reductionist, and that instead of looking at the individual proteins, knowing that the essential amino acids were the key, because called essential because human beings can't produce them, right?
Right.
Unlike the
Yep.
dog you just heard barking, human beings have to take them in a diet. So I think that's what we look at from a very reductionist standpoint. Is like if we just start with these EAAs, what effect do they have? And we actually started. This was just probably 30 years ago now, because I remember doing the work in the laboratory where I took skeletal muscle, hydrolyzed it, ran it for the EAA profile, and then our first experiments were actually mimicking the composition of skeletal muscle. Now, it worked, yes, but then we started realizing, well, it doesn't exactly work as well in aging. It doesn't. So over those 25 clinical trials, to make a very long story short, we ended up tweaking various aspects of that formula and ratio of amino acids that gave us a maximal response. So everybody always asks me, well, not everybody used your formulas, the amino acids work, etc. In a general sense, yes. But what I can say is that our patents were derived from these 25 clinical trials, the iterations and tweaking formulas. I can say that I have the data that shows that they were beneficial. Okay. That's where a lot of people can't at this point. So I'm not
Right.
dissing anybody else's formula or saying theirs doesn't work. I'm just telling you how ours was derived
Off
scientifically,
data. Off
empirically.
data. Yeah. Yeah. Yeah.
So, yeah,
One thing. So you mentioned your shoulders. Your shoulders good. You did some boxing recently.
last night.
Yeah. Maybe you could set up a Mike Tyson fight. What do you think about that?
Hey, dude, and I turned 68 in a week. I got no illusions. I'm just, like I told him going into this, I wasn't functional. I just want to be functional again. You know what I mean?
Yeah.
I just
Yeah.
like the training. I have no illusions that I'm going to lift heavy again or spar heavy again or any of the above. But I just like the activity. I like, I like martial arts. I've always have been involved, and I like the human chess match. I like training with these guys. They're all like much, 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 much better than I am or will ever be. It's just like on that note, it was like being with Bob Wolf in the early years.
Mm,
You know what I mean? It's
mm.
like here I have an intellect that is far greater than mine. I have a scientific experience that was far greater than mine. It's like anything else. If you want to be a world-class athlete, you got to train with world-class athletes. I've always believed that, right?
Yeah.
If you want to
Right.
get better at X, you got to train with people who are better at X. That's why all my martial arts and boxing partners are much better than I'll ever be. They're half my age or more or less. You know what I mean? One is in his 20s. You know what I mean? So again, they're just nice guys who I train with and get a lot out of, and they're nice enough to keep me alive. You know what I mean? They can pull the
Yeah.
kicks. They can do the things they
Yeah.
need to keep me
Yeah.
out of the ICU. I mean, I'm under no illusions, like I said. So it's just great to be around people like that. I'm very appreciative.
Yeah. Yeah. I can relate to that a little bit. I played basketball when I was younger, and I didn't play basketball for like probably like 15 years. And I started playing last year at the rec center with the students. So after having not played or doing any sort of cardio consistently for 15 years, it was a tough learning curve, but it's fun. It's fun.
Well, that's important, right? I mean,
Yeah.
as you age, activity is everything, and that's why I said I just want to keep doing the activities that keep me highly functional and enjoyable, and like yeah, I'm under no illusions. Like you said, no Tyson fights, etc. So yeah,
All right. Let me ask you. I want to ask some specific physiology questions and protein metabolism questions, but I think we're at dinner in Medellin, and you were telling us a little bit about the bed rest
Ed,
studies. Yeah, and I remember vividly in my head you had a nice description of the subjects of the volunteers that would volunteer for this study, and then you had a specific I don't know, a specific hemodynamic adverse event.
oh man, yeah, wow. I got to
Not
be careful
you,
what I
but
say, I
one
guess.
of the subjects.
Yeah. So At that time, the bed rest studies, and I lost count. I've done a number of them throughout my career, had a number of grant projects. In fact, I had NASA funding like for the first, 20 years of my uh, career. And NASA used bed rest as a ground-based model for the muscle loss. And there were some, if you did like 6-degree head down tilt, the hemodynamic changes were similar to space flight. So in those early years, we combined all that. But later on for me, it's just the fact that the inactivity led to muscle loss. And the mechanism we found, in fact, it was my 96 paper, was the decrease in protein synthesis. So again, things we've taken for granted, Kurt, over all these years. I keep telling the younger guys in my lab too, because they throw out all this stuff, but you have to keep in mind that in the early days we didn't know what the mechanism was. That was the whole idea for going to Bob's lab, learning protein kinetics, learning tracer methodology. Like, where was the problem? Was it too much protein breakdown or not enough protein synthesis? The bottom line is not enough protein synthesis. And now teleologically it makes sense, because if you're not using the muscle, there's no requirement to have new proteins being made, right? Like just the fact that you got on the basketball court last night screwed up a whole number of muscle proteins that have to be replaced, right? Well, if you're laying in bed, that doesn't happen. So protein synthesis goes down, and that's the reason for the muscle loss. And in 1997 it was, I did the same thing in space flight. And John Glenn, if you remember Senator John Glenn, he was one of the subjects. And that's exactly what we found through some methods by looking at N15 alanine, whole body turnover, and 3-methylhistidine, labeled 3-methylhistidine excretion, things of that nature. But the bottom line, it wasn't protein breakdown, it's protein synthesis that stopped. So, yeah, they taught us a lot in that regard. And so that's why, again, if you just go to, 25,000 feet, which is the 30-some year career, now you see why exercise becomes important. Now you see why aging becomes a problem when, activity and everything else diminishes, because now you have what I call layered physiology or layered problems. So you get older, the muscle becomes more resistant to the effects of amino acids, protein, amino acids, whatever, becomes more resistant to that. So for a given amount of amino acids, in fact, we had to that point, we had a postdoc named Christos Katsanos, who's actually doing very well out in Arizona. He's a professor now, funded. He's done a great job. But his study with us showed that for a given amount of amino acids, I think it was 7 grams, for example, the young subjects had a pretty substantial response. The older subjects had a blunted response. And that's what led us to realize that we needed more leucine, because when we upped the amount of leucine, then the older had the same response as the younger. So you see what I'm saying? It's like, I don't have to tell you this because you're a physiologist. Physiology changes with aging, right? And so what we have to do is learn what those changes are and learn how to adapt to them. And so that's kind of back to the earlier point about how our formula and everything else evolved. Well, that was one of the aspects of it. It's like we realized that the way the formula was constructed, the older people weren't getting what they needed out of it, weren't getting the same metabolic response. So we had to tweak it accordingly. So that's kind of how things were derived and over the years. And, when you combine the resistance of aging muscle and then the inactivity and, on and on, you have these series of problems, like hurdles that you have to overcome for health. And what they don't realize is that most people don't have the wherewithal or desire to do that. And pretty soon you're type 2 diabetic, and now you're really in a bad downhill slide, right? Diabetes, heart disease, cardiovascular disease, kidney disease. Oh, Ozempic will take care of that. Don't worry. So, that's the problem. I mean, if I have an axe to grind, it's like our whole system. It's called the Food and Drug Administration, but it's really drug administration. Nobody gives a shit about food. And so, that's what we have here, is that the pharma can solve every problem known to mankind. And as you and I both know, it may solve, and it can solve some problems. And I'm not totally dissing farming. Like, where would we be without antibiotics? On and on and on.
Mm.
But the fact that you can continue to abuse human physiology and farmers going to step up for you is a naive fact. You know what? I may have said this to you before. I just firmly believe this. Like, everybody chooses their way to die, right? This has a timestamp on it. We all know that. Okay. And throughout your lifetime, you will do things that, in essence, will choose your way to die. Okay. Like, you could argue that I've kind of, run out of the brake pads, right? I've been going hard at athletics and everything else. And so now I've had two body parts changed out. You know what I mean? So you could argue that, which is true. I brought this on myself. I never complained about it, but I knew damn well that this is where I was heading. You know what I mean? If you want to do these things in your life and have this kind of activity, you want to jump out of planes, you want to do, you want to rappel, you want to do these, eventually you're going to pay the bill. Like the old saying, my dad used to say, you want to hear music, you got to pay the fiddler, right? And I'm paying the fiddler. But I don't mind that because I understand that. But the other side of that is you've done nothing and you've developed these diseases, and these diseases are much worse than an orthopedic surgery. I've had, I don't know, I lost count how many orthopedic surgeries I've had. I'm still functional, still doing fine.
Yeah.
It's just surgery. It's just surgery. But my God, these disease states are irreconcilable. I mean, once you get heart disease, there's only one heart you get. You know what I mean? Once you get kidney disease, you got a miserable way to die. Is the way I look at it.
Yeah. Yeah.
Lecture over. I'm sorry. I didn't mean to get on that. But, I just, at this point in my life, I've seen so much of it and heard so much of it that, I mean, people would have to understand.
Yeah. Yeah. There's a lot there.
Yeah, no, there was a lot to unpack on that one. But I mean, the bottom line is that, the other side of that, Curtis, there's enough information out there now, since your time and everybody comes. I mean, like, if you had to ask any professor in exercise physiology, like, what's the best way to go about this? I mean, most people in a general sense would tell you activity of some sort. Nobody says you have to run 10 miles a day or lift 4,000 pounds a day. Activity, consistent activity over your lifetime, and an awareness of the diet. You can't eat Twinkies every day of your life. You can't have French fries every day of your life. And oh, by the way, I'm going to grind one more axe, Kurt. I'm sorry. You just
no, go for it.
You opened up the floodgates, brother, on this thing. So I freely I've been transparent in all my work and all the research, and we do a lot of work for commodities. In great part because our research has shown over the years the quality of protein is much better than plant protein, okay? That's the data. The bottom line is you need quality protein sources. We have shown it we and others, I'm not the only one, as you know, have shown it in every circumstance that quality protein sources are better for people. And this is where all the epidemiology and the NHANES and nurses studies and all those things went wrong, is they showed this correlation over the years of, they call it red meat, okay, which again is a misnomer, right? A lot of meats are red, and cardiovascular disease. But what people don't realize is, in fact, I may go to lunch with friends here now. If I go have a burger, what comes with the burger? Fry.
Yeah.
Roger that. You know what I mean? Yeah. They never accounted for the fact that, all this, cheap carbohydrates come with it. Let's face it, those fries are very unmeaningful to the metabolism physiology, right? They're just dead calories for the most part. You know what I mean? It's not like they serve any great importance. It's not like we were carbohydrate depleted or something, you know what I mean?
Right.
So that's the problem you have over all these years, is like you've focused on one thing, you've ignored the ancillary things that are contributory, and you come up with the conclusion that animal proteins are bad for you. Well, they're not bad for you. And again, I'm going to use a teleological argument. How did we get here? We were hunter-gatherers for thousands of years. How did we get here, right? We hunted the game, we shot it, we ate it, we lived. Come here, sweetheart. So, that's why I keep saying, it doesn't make sense, and it still doesn't to this time.
Well, kind of to that point, and I saw David, DC David Church present this at least one or two times, maybe once, and maybe I've just seen it online. But that study with the burger patty, the plant burger patty versus the traditional beef burger patty.
Yeah. It was the Impossible Burger. It's the
Impossible Burger.
Yeah.
Impossible Burger. Yeah, yeah. If I recall correctly, it was you'd have to eat 2 of those Impossible patties to get the muscle protein synthetic response of the beef patty, which someone could do, but if the concern is, say, metabolic health, obesity, insulin resistance, then that I don't remember the caloric content, but that increases
almost twice. It was almost twice the calories. You're right. That's the point.
Compared the Impossible Burger to a beef burger, and we measured the muscle protein synthetic rate. So originally, we were just going to do like a 4-ounce beef burger versus a 4-ounce Impossible Burger. But I thought, we're going to get in trouble because the beef burger has more protein and more essentials than the Impossible Burger, and people are saying we're just going to be biased here. So we actually added on a double Impossible Burger group. So they got 2 4-ounce patties. Beef burgers got about 25 grams of protein and about 10 grams of essentials. The double group actually has like 15 grams of essentials and like 36 grams of protein. So the double Impossible Burger had 50% more essentials than a single beef patty and almost double the amount of protein, but it gave you the same muscle and whole body protein synthetic response. I also measured the plasma essential amino acids, so the amount that you eat and then the amount that gets out in the plasma, right? And I was shocked that the Impossible Burger, the 2, everything I'm talking about here is the 2 Impossible Burgers. So 8 ounces of Impossible Burger had 50% more essentials than the 4 ounces of beef. And despite having 50% more of those essential amino acids, the concentrations of the plasma essential amino acids were lower than the 4 ounces of beef. And that's to your digestibility point.
So that's what that study, I think, is the crux of the entire issue, is that, and I say this all the time, you probably heard me say it when I gave the talk, I got nothing against vegetarians, vegetable proteins, I got nothing against any of it. But again, the data are the data. It's like, if you're concerned about muscle health and metabolic health as you get older, you would go with the bang for your buck. Mm-hmm. If for 100 calories, I can get X response, and then for 300 calories, I get X minus response, why would I do that? You know what I mean?
Yeah.
Why would I take the 300 calories? I mean, it's just something you and I have to work off in the gym at some point. I mean, they're not free, So, yeah, so I mean, it's a very important issue. It's a very important issue, and I think people miss that. And the problem with a lot of vegetarians, it becomes again a religious or cult thing to them. Fine, you do you, but my point is you can't ignore the data. The data are the data. Right. Whether you choose to believe them is your problem, but the data are the data.
Yeah, well, and I think you mentioned if you're worried about muscle health, and I don't want to mischaracterize people, but I think a lot of people, especially those who might go for the plant-based, vegan type of diet, is they may not understand the importance of muscle health and the role that it plays in overall, age-related health, cardiometabolic health, and they might be preoccupied with, some other aspects of health, but not understand the importance of keeping skeletal muscle healthy and functional and all of the downstream implications that dysfunctional skeletal muscle could lead to.
That's exactly right. And the bottom line is this, it's too much carbohydrates. I think that's what people miss. And our society is inundated with cheap carbohydrates everywhere you go in the grocery store. And the reason for that, by the way, is because carbohydrates have a shelf life of forever. In fact, that's the problem with the military rations. It's easy to put carbohydrates into anything, but it doesn't give you the metabolic response you want. Now, if you're out in the field in Afghanistan on a mountain, calories are calories. I mean, you don't care at that point. But again, to maximize the metabolic and physiological responses and resiliency, carbohydrates don't get you there. And in fact, we have a recent paper out that showed you again, back to those military studies, Jess Gwynn from USARIUM is the first author, and DC, I think, is the second author on that. But it shows you that what we were looking at is, okay, we provide this in a hypochloric situation, like where do the carbon skeletons come from? That's what kind of the question we answered. Do we need, can the carbon skeletons come from more EAAs or carbohydrate? The answer was the carbohydrate didn't do the job. Okay. So it tells you again,
Right.
I didn't measure fatty acid storage and I didn't do fatty acid kinetics, but I can tell you from experience that those carbohydrates were predisposed to being stored, not utilized.
Right.
So again, and that's why I say the problem with a vegan diet is there's just too much carbohydrates there. The body is meant to take in, more protein than people give it credit for.
Right. Right. No, that's the point that I mentioned with for students. People might look at the exercise or athlete recommendations of carbohydrate, and they might think, oh, I'm doing a 30-minute spin class or a 30-minute Zumba class. I've got to eat 5 to 7 grams of carbohydrate per kilogram, and to your point, what I mentioned is most people, non-athletes, they're not depleted of glycogen. They're full of glycogen all of the time. The issue is not enough carbohydrate. Most people are top to the brim, if not more carbohydrate than they need. And I think that's probably the case.
And even to your point, look at the athletes that have to worry about carbohydrates. They're extreme. They're marathoners or more than they're tour guys, you know what I mean, who are cycling for 6 hours. I mean, those are extreme forms of exercise. And like you said, how many people do that? So yeah, to your point, you just don't need that many carbs. And that's a fallacy, again, perpetuated by these manufacturers and supplements and all that stuff. It's just not required. Just not required. And in fact, I think it's doing them wrong. All these people that you and I know that don't have degrees in our area, they're, like you said, you walk in the gym, they're hammering themselves away, and yet it's one step up, 2 steps back, right? Because how little that 30 minutes of Zumba actually consumes in terms of calories, right? Let's say 200 calories, give them the benefit of the doubt. 250, 300. But you just ate something that gave you 300 calories. You know what I'm saying? You ate that whatever power bar, whatever bar that is, that had 150 or 300 calories. So where have you gotten on that?
Right.
Yeah, it's they just don't understand simple metabolism. I'm sure in graduate school, like all of us, you measured the cost of exercise, and you realize how little that is in terms of calories.
If you're not devastated after you learn that, you didn't learn it.
That's right. That's the hard, cruel truth of it. Exactly.
Yeah. I do want to ask you. We've talked about the plant-based protein versus animal protein, high-quality protein versus low-quality protein. Can you give just like a little description of what we mean, or what is the difference between plant versus animal, which would make the latter high quality and the former of lesser quality, and the role and importance of essential amino acids in that?
Yeah, so it has to do with two things. One is simpler. It's the presence of all the essential amino acids, okay? A lot of the plant proteins are deficient in lysine, for example, okay? But more importantly, what we've come to find out, there's an older colleague named Paul Maughan out of New Zealand, and Paul and Bob have gotten to be, because Bob lives in New Zealand now, so they've gotten to be very good scientific colleagues with us. And Paul, over the years, has carefully characterized total ileal digestibility. In other words, what makes it out of the ileum into the circulation. And the point is that the plant proteins are not as highly digestible as the animal proteins. So there's another problem you have. So even if you married up, okay, let's say animal, plant, 25 grams of protein, you're not going to get the same peripheral amino acids. Because the 25 grams of plant protein is not digested as well as the 25 grams of animal protein. So your peripheral amino acids, your circulating amino acids are not going to be the same, which means that your response of protein turnover, both in the body and muscle, is not going to be the same. That's the real take-home issue that people don't get.
Right.
That you can't get away from. Again, I didn't design the plants. I didn't design the physiology. Don't get mad at me. I'm just telling you that's the data, okay? That really is the crux of the issue there. Now, the question I've always asked, which I could give you my hypothesis, but nobody's ever funded me to do it, is let's say you took 25 grams of plant protein and then you gave X grams of free-form amino acids on it, would you get the same response? My hypothesis would be probably yes. Okay, but you see what I'm saying? Here's what people don't get. The skeletal muscle doesn't know where that phenylalanine came from, doesn't care where that phenylalanine. It could come from sigma, it could come from a deer, it could come from soy. It doesn't care, okay?
Right.
It's just like, it's totally biochemistry. It's like, I need this substrate to make this protein, and that's it. That's what it's looking at. So that's the point. It's like, how am I best getting that substrate to where it needs to be utilized? Okay. A lot of it's taken up first pass in the liver, right? In the portal vein and all that stuff. So that's another thing you have to think about. And rightly so, because the liver is the most important protein making organ in the body. So again, what's left and what gets out to the periphery is what the muscle uses. And so, you have to kind of think of it that way in terms of like more amino acid kinetics dynamics than like, source, et cetera. But that's very important. And that's what people don't really seem to understand, and the difference between the two. And again, because they get caught up in this cult-like religious fervor on it. And fine, you do you. I don't care. I'm not telling you not to be a vegetarian. You do what you want to do, but I'm telling you that the data shows here's the reason you have to be alert to the I guess side effects, if you will, or of being a vegetarian. Okay,
right.
That's not a proper term, but I mean the additional considerations. That's a better way of looking at it.
Right.
Additional considerations for being a vegetarian.
Yeah, definitely. Can you speak a little bit to the muscle protein synthesis methods that you used, the tracing methodology? Because I think, even for me, I've never done it, and I read papers, but don't have a full, like, super detailed idea of it. And I do talk about it in classes and try to explain it the best I can. But can you explain how that method works and how it gives us an idea of how those amino acids are metabolized and where they end up?
Yeah, so I'm going to give you a simple explanation that you can use, because the rest of it is really heavy dork stuff. You know what I mean? If it get into your eyes, you're going to glaze over and you'll probably hit your head on the desk.
Okay.
So
I did have to take chemistry 3 times at junior college, so yeah, thank you.
So, well, more importantly, it's kind of like the analogy I use is imagine having a chem light and you're going through a dark room and you can follow that chem light to wherever it goes. Okay, that isotope or tracer is that chem light. So you introduce it into the body either by infusion in the old days or orally for deuterium. It really doesn't matter. The idea is that once you get to what we call steady state in the precursor pool, so let's say we used phenylalanine was a common tracer that we used. So we'd infuse it or whatever, we get phenylalanine to steady state. So that means that the movement of that tracer into muscle and the presence of that tracer in the muscle, or the light, if you will, will allow you to calculate how rapidly the muscle is being made, okay? Because it's taking from this pool, the blood, okay? The extension of that is the dilution of that pool tells you how much the muscle is being broken down, because it's unlabeled phenylalanine from the muscle that's breaking down and diluting the label. And so in our dork speak, we call that tracer to tracee ratio. It's just enrichment, or the amount of that tracer in the plasma or the precursor pool, whether it's plasma, intracellular, if you're taking a muscle biopsy, whatever. So that's the simple explanation of this. That's why it in a general sense works well. Now, I will tell you that over the years, the methods for determining synthesis are much better than the methods for determining breakdown. And we have never been able to circumvent the fact that the breakdown methods are only highly reliable in the fasted state. So we've never been able to really do it well in a post-absorptive state. Now, we have surrogates like 3-methylhistidine label and things of that nature, but they're more qualitative than actual quantitative. In the early days, we were able to do pretty elaborate work with in burn patients and everybody else in Galveston. We cannulated the femoral artery and femoral vein and took muscle biopsies during tracer infusion. So we actually, if you look at the earlier papers, we actually were able to assess the kinetics of these tracers from the blood to the intracellular to the muscle protein, and the other way, arterial venous shunting, the whole 9 yards. So we got a good methodology of a good encapsulation of how the amino acids move given both fasting and post-absorptive states. So to recap, like I said, it's very easy to get bogged down in dork speak, but to recap, it's just like following this label, which is an isotope, a stable isotope, but it's following this label with this light, or however you want to look at it, through the body, and from that being able to calculate based on certain assumptions and certain things, synthesis and breakdown of skeletal muscle.
How did, and to be honest with you, I'm not sure of the timeline because I was not super familiar with a lot of this type of muscle protein synthesis physiology until I got really into my PhD. But how did that growing field of muscle protein metabolism and being able to do that tracing and seeing the dynamics of that and the kinetics, how did that coincide with the growing understanding of like the molecular physiology of, say, mTOR signaling, all the ribosomal biogenesis, like all that signaling pathway? Was that sort of a parallel fields?
Somewhat. That whole mTOR thing was kind of developed in Jefferson and Lee's lab at Penn State many years back, and then promulgated forward. The interesting thing that I will tell you now after all these years, so think about this. What we were describing is, like, we call it mixed muscle FSR, like a gross measure of protein synthesis. And so the interest was, well, where's this occurring? What's more important, et cetera? And in the early days, we separated out fractions like myofibrillar, sarcoplasmic, and mitochondrial. And the bottom line is that when there was a stimulation in all three. So after a while, we just stopped separating all that. It was just more work for nothing. So it just seems like when adequate precursors were available, all muscular biology took advantage of it. Okay.
Yeah.
So I mean, everybody looked at these signaling molecules and all that stuff, but if you really look at that literature, there's no rate limiting step there. And in fact, it's permissive. And in fact, Bob has written things on this very nicely and eloquently in recent years that shows that the changes in the translation initiation were not related to synthesis at all. So in other words, you can have a stimulation synthesis and it doesn't change, or they're very temporal. They're very limited temporally, right? Because you have this long chain of a translation initiation. If you don't catch it quickly enough or at the right time in the biopsy, it never showed up. So I think the way to look at it, it's describing permissive biology. You know what I mean? As you know, human biology is rarely limited in like one enzyme, one, you know what I mean? And here's what I give as a good example. Marcus Baumann, excellent scientist, physiologist. A number of years ago, he did a training study with young and old subjects, 16 weeks training, hammering them, et cetera. Did all the translation initiation, all those signaling, et cetera. And one of the things he found in his paper is that between age groups, the the point in the pathway was different. In other words, like, for example, young, right? Maybe, for example, right at mTOR, that lit up. But an older person down here at AKT, you see what I'm saying? So
Yeah.
then
Yeah.
that tells me that biology is permissive in various ways.
Right.
However, the way I look at it, again, going to 10,000 feet, is all that is irrelevant if the substrate isn't there, right? So
Right.
if the essential amino acid precursors aren't there and aren't there in sufficient numbers, all that's counting angels on the head of a pin. I'm not saying it's, it's garbage science or it doesn't mean anything. That's not what I'm saying.
Right.
I'm just saying that that's why when you put all that in a big barrel and look at it, I mean, the measures of raw synthesis and raw breakdown tell you what you need, right? It tells
Exactly.
you in a balanced sense whether the body's in a positive balance or a negative balance. Does it matter whether that's AKT, P76S kind of, you see what I'm saying?
Yeah.
This is Bob's term. He used to me many years ago, and it's always stuck with me because after a while, I started really realizing what he meant by it. It's counting angels on the head of a pin. You know what I mean? I mean, it's not really answering the entire question. It's answering a question, but not the most important one.
Yeah, no, that I really take that point because, I don't know if you are on YouTube or podcasts or, social media to any degree, but, and there's a lot on health, fitness, training, nutrition, longevity, and a lot of it, there's some good stuff that is pretty rigorous in science-based, and then there's a lot that is sort of masquerades as science and use terms and terminology kind of performatively and kind of to obscure things and make it sound like they're talking about something novel and, cutting edge and scientific and whatnot. But in reality, they don't know what the hell they're talking about. But yeah, to that point of, and I make this point to students is, you can't take something that happens in the cell, some molecular marker, some cellular marker, some cellular process, and then translate that or extrapolate that into some actual clinical, physical, real life outcome or take any real sort of meaning to that.
I agree with you entirely. And what you find in those things, which I don't pay attention to, is that there's not an understanding of physiology. Because I still got my friends, sending me things. What do you think about this? They'll text me this link. What do you think about this? It's just that it's the lack of understanding of physiology. So either the person or people selling that don't understand it or don't care or taking advantage of the fact that most people don't understand it. That's just not the way physiology works. By the way, it's never that simple and it's never that easy. Okay. I tell everybody it's not that freaking easy. If it was that easy, would we have a 60% obesity rate in this country? No. Okay. It's just not that easy. The bottom line is you got to pay the price, baby. You got to pay the fiddler to hear the music. You got to pay the price. You got to eat well. You got to eat the right things. You got to be active. It's just that simple. And, back to the activity, like I keep saying this is like, again, look at the exercise literature, Kurt. Again, exercise in any clinical pathology, always beneficial. Always beneficial. But nobody does it. I've done studies myself, like Marcus and I had an aura once years ago looking at exercise after joint arthroplasty, knee and hip arthroplasty. And the exercise regimen was completely, was absolutely beneficial. Of all those people we had on the exercise program, there's a onesie-twosie that maintained exercise after that. It's just that people don't do it. People don't do it. That's why the NIH stopped funding a lot of that stuff. It's like, we all know it's beneficial, but they're not going to do it. That's why they're obese. You see what I'm saying? If exercise was a part of our life, if JFK was successful in the 60s, long before you were born, in promoting, health and all that stuff, it's gone from the school anymore. That effort he had, which was a great effort in the 60s, just died of natural causes. And this is what we have. What you described to me is the fact that this field is ripe for snake oil salesmen because people want quick answers. They want any answer they can think of, like that allows me to sit on my ass, drink 3 drinks a night, eat burgers and fries, and gain weight and still be fine. And it just doesn't work that way. And again, that's not Arny being an asshole. That's the data. You know what I'm saying? It's just that simple.
I do want to ask you one more physiology research question. I'm sure you saw the paper that did, I think it was out of Van Loon's lab, where they did the 25 grams versus the 100 grams, and they looked at a lot of things, but including the muscle protein synthetic response, and then actually the amino acid accumulation in the skeletal muscle, if I recall correctly. Now, typically or generally, and this is maybe an oversimplification, but the idea was that, once you consume a sufficient amount of high quality protein, on the whole protein end, 30, 40 grams, on the essential amino acid end, I think it's like 6 to 12 grams. That's going to
15,
elicit
yeah, that's what we found.
12 to 15.
Yeah.
Okay. That's going to that's going to be a threshold for muscle protein synthetic response. And that will, over the time course of 3 to 4 hours, or 4 to 6 if it's consumed with a mixed meal, and then there's a refractory period. But the general idea, and this could be mistaken of how I sort of took that, is that there was a response and then a refractory period, and then any additional protein intake didn't contribute meaningfully to the muscle protein synthetic response. However, this paper, they had one group 25 grams, the other group 100 grams. And what they showed over, I believe it was 12 hours, was a much more, not a higher peak, but a prolonged muscle protein synthetic response over those 12 hours with, I wouldn't say a proportional greater increase of amino acid accumulation in the skeletal muscle, but a greater accumulation nonetheless. So for you, did that sort of change anything in terms of our idea of how protein metabolism in the time course sort of worked? No?
No, I think it's quite consistent with it where everything, the data has been. I mean, Luke's done very nice work over his career and continues to do very nice work. But I just think that it's just consistent with everything we had found in the past. It's like that 100 grams was a pretty large load. Again, takes a lot of time to digest, puts a lot of amino acids in the system. Most of those will be used for whole body turnover. I don't remember the minutia of the data because DC and I discussed this, but I know whole body protein turnover was a lot more favorable with a larger dose, which makes sense. The muscle itself, as you adequately described, will stimulate, and it won't stimulate forever. And by the way, in Galveston, we tested that exact asset. There was a fellow we had here was an anesthesiologist from France named Julien Bouy. And Julien, Mike Rennie at the time, he was did a year stint with us, and Bob did this project. In fact, I did the biopsies on that project way back when we infused amino acids. We infused them over time period. And we showed the stimulation, and then, like you said, there's a refractory. There's only so much stimulation you can get, okay? Right. And if you're going to stimulate further, it usually entails a stimulation of breakdown. That's what happens in a burn patients, by the way. If you just measured synthesis in burn patients, you'd think they were anabolic because the synthesis is twice as high as a normal individual. The breakdown is 3 times as high.
Okay.
So that's the missing piece of that puzzle that's after. So yeah, it shows you that it's the regulation of extracellular amino acids that do that. Okay, not intracellular. And that paper nicely ironed that whole thing out because we looked at FSR, we looked at intracellular amino acid concentrations, we looked at extracellular amino acid concentrations. And the intracellular amino acid concentrations did not increase until you kind of flooded the muscle at a very high dose. So again, it's consistent. What he found was consistent with everything we had found before, that you keep providing the precursors and there will be protein turnover in the body, but the muscle itself is not ad infinitum protein turnover. I mean, we all know that, right? I mean, you and I eat a lot of protein. I weigh less than you. You know what I mean? I'm not as stout as you are. You know what I mean? It's just that the human body has its limitations. So yeah, at some point you got to realize that there's going to be a limit to the synthesis and or anabolism. and other things have to occur to balance that out. So, I think his data was quite consistent with the literature, and I just think it was a good way of showing what we're used to eating, and then let's go more extreme. What
Right.
does that do? 100
Right.
grams is a lot to eat, as you know. That's a pretty
Yeah.
substantial steak.
Kind of, we alluded to them earlier, but maybe just to focus on them a bit more. The timing significance versus the overall amount significance from a protein and essential amino acid standpoint. Just generally speaking, how much should one consume? There's been a couple of papers that have come out that sort of, touched on that. I know there's a little bit of back and forth regarding that as far as like what is an upper limit. And then I think an important one from a practical standpoint is that people might have concerns on are higher protein diets unhealthy, right?
Okay, so timing. Yeah,
Yeah.
let's talk timing. So let me refresh you and listeners that the approach I take is fairly reductionist. I mean, from EAA standpoint. Okay, I mean, so all the work that has kind of led us to this point that it's really EAAs that matter metabolically, physiologically. Okay, that's why we take in protein, right? They're the nitrogen carrying component. We can't make it. We got to get it. So putting that all together, it really matters the amount of EAAs in the blood at a given time. And I say people this all the time because they get wrapped around the axle. The body doesn't care where those EAAs come from. It could come from peas, it can come from steak, it can come from Sigma Chemical Company. It doesn't really matter. Okay.
Right.
So, but what does matter is like how fast you can get them through the digestive process and out to the peripheral blood, because that is the signal for protein turnover, both at the whole body level and at the muscle level. So that's what I think people miss all the time. And that's why you got this big argument about this protein versus that protein, et cetera, et cetera.
Right.
And as you well know, it's about calories too. You can't eat protein ad infinitum without consuming X number of calories. Okay, so you have to understand that the body deals with that protein in a given format, and when it can't deal with it, it has to excrete it, right? Because nitrogen ends up being, or nitrogen products end up being toxic to the human blood at high enough levels. So we get rid of it in forms of ammonia, mostly urea, okay? The urea cycle. Okay, so now given that, in terms of timing, again, I always refer to this because my colleague, who is now running a lab, Dr. David Church, had put out a nice paper that he's going to add to here in the near future in 2020 in Nutrients. And he nicely showed through a wide range of EAA intakes, everything from a mixed meal to like beef to like whey protein to like free-form amino acids. So the whole gamut of intake forms clearly showed that there was a fairly substantial relationship between the EAA area under the curve and also Cmax. In other words, the maximal concentration and time to Cmax. Okay, so without boring you or being too big of a dork, the bottom line is that the faster you can get them out there and the more you can get out there, the bigger the response in protein turnover, both at the whole body and the muscle level. Okay. Again, that's a take-home message. The more you can get out there, the faster you can get them out there, the better off you are. Now, in terms of timing, we did a lot of work many years ago looking at timing in terms of amino acid ingestion and exercise, which I know your audience is exercise oriented. And the bottom line there is that if you take in an amino acid source, let me just be more generic in that regard, like within an hour prior to resistance exercise, the effect is quite interactive. And the reason the effect is quite interactive is because blood flow is dramatically increased to the exercising muscle. So it's bringing all those essential amino acids in contact with the muscle membrane. Okay. That's really the key to that. And again, I refer people to these papers, but it's before, any of them were born or even, but in the early 90s, we had a fellow named Johnny Biolo, who's now, an old professor in Italy, in Trieste, Italy. And he did some outstanding work in Bob's lab in the 90s and showed we had some pretty elegant methods. I don't know if you remember that. We did, femoral femoral catheters, excuse me, and a cross limb balance model. And so we had some pretty elegant methodologies back then. And from that, we could clearly show that the blood flow dramatically increased the amino acid delivery to the exercising muscle. Now, we also showed that you could get an effect by consuming the amino acid source after exercise, but it's not as great as before exercise. So what's the lesson there? Well, the lesson is two-part, and I haven't talked about the second part yet. The first is delivery of amino acids in conjunction with increased blood flow during exercise. However, I'm an example. Like I can't take in like whey protein or something before I exercise. If I do, I'll see it in 3 to 5 minutes. I can't digest. You know what I mean? A lot of people can. I've seen people drink it and then had it. I mean, but I can't do that. A lot of people can't do that because
I won't ask you in which direction you see it.
well, yeah, I know it comes out where it went in. To be honest with you, and it's just because the digestion takes time. That's where free-form amino acids have a dramatic advantage because they take no digestion. They simply, move through the GI and into the blood very quickly and get a tremendous response. So that's why in my lifetime I've switched entirely to that. And in fact, I got them right here, and I'm going to go work out after you and I are done, and I can do the same thing, just drink them while I work out. So if that's not an issue, then fine. But those are two things that you got to kind of keep in mind. So again, take home message: within an hour before the exercise, and or within an hour after. And as you know, because you're an exercise guy, it all has to do with exercise intensity, right? If you get intense enough, there's nothing you're keeping down, right? So if you're doing repeat 100-meter sprints or whatever it is, I mean, it's kind of irrelevant. There's not much you're going to keep down. But usually when we train weights, we're far below the 80-some% threshold, and it works fine. Does that help you and answer your question with timing?
Yeah, and I think also to bring in a practical strategy is the idea then to sort of match the next, say, protein intake with the expected time course of that muscle protein synthetic response of, say, 3-4 hours, or maybe even more if people are consuming those amino acids, in food form, with other macronutrients as well?
Yes, but I should have said this because everybody is, I realize over the years, and I've been talking about this, people get confused, and rightly so. But as you know, human beings have been living for thousands of years by eating protein sources, right? So it's obvious that's required. But what I'm talking about in our field of exercise and is optimization of intake. That's, I think, where you really have to make the distinction. Can you make gains and do this by training and just eating? Yes, you can. But if you want to optimize it, or if you want to overcome resistance as you get older, and it's there, baby. Trust me, it's there. You need to do these. You need to pay attention to the aspects I was discussing. How you're delivering those EAAs, how quickly they're going to get out there, how high they're going to raise. Because one of the things that DC showed in his paper is you really need about 100% increase in EAAs to get a significant synthetic response. Now, does it happen less? Yes. But again, I'm talking about an optimization. So it kind of fits into your second question about what the amount is. And so, again, the amount is not a straightforward question. Now, Stu and a lot of people with Tipper and all those years, the groups, Phil, Atherton, they've done some very nice work, and everybody kind of comes up with the mantra about 25 to 30 grams of whey protein. However, the reason that was derived is because whey protein is fairly highly digestible. And again, the EAAs get out there in a fairly quick manner. And that's why that protein has, in our lifetime, and rightly so, gained so much attention. However, the formula changes entirely if it's not whey protein, to be honest with you. So, if there's a highly digestible and a high quality protein similar to that, probably so. But if it's lesser, certainly certainly plant proteins that we've investigated, it's going to take more than that. And it's going to take longer because they're less digestible. And Paul and his group have done a great job at looking at this, because everybody's worried about like how much amino acids do we need on a daily basis, how much protein, the whole WHO, RDA garbage. But his point is this: it's like it depends on how digestible it is. And over the years, he and his groups have done a very good job at looking at total ileal digestibility. We call it TID. And that makes all the difference in the world. In other words, it doesn't matter how much amino acids is in that protein when you send it to the lab. It matters how much is making it through the gut to get to the periphery. Okay, that's really the point people are missing. So that's been kind of a mission profile, if you will, of Bob and Paul and those guys over the last several years to show that there are no ounce equivalents. That's the biggest lie they tell. There's no ounce equivalents. They're not equivalent at all, and you have to attention to the protein form and format before you can make. And also, as you know, let's say, in fact, DC again, first also beef versus a Impossible Burger, which is kind of mostly a soy burger, and of course it's marketed as a replacement, et cetera, et cetera. Patty of beef, patty of Impossible Burger, and then 2 patties of Impossible Burger. Okay. And we showed that the beef versus Impossible had a much greater effect on muscle and whole body protein turnover, okay? And the amino acid levels. When you gave 2 Impossible Burgers, you had a similar effect, but you gave twice the amount of calories.
Right.
Okay. So again, another conveniently missed, thing that people always say. And it goes back to what I was telling you. The body doesn't care where the amino acids come from. All it sees is, hey, there's a physiological or metabolic signal here. I do what I need to do with it. I replace all these proteins that need to replace, which as you know are thousands on a daily basis. So that's an important point and an important way to distinguish. So back again, so I don't obfuscate this whole thing. When you talk about amount, that has to do with the quality of protein and the digestibility of protein. That's what matters. So that's what, again, the public misses. And of course, all the people who manufacture and source it are spreading, information, whether it's right or wrong, in marketing format, and you get all these numbers and better than, just as good, whatever.
Right.
I'm telling you from a scientist and a scientific standpoint that's the important thing you have to think about is the digestibility, which translates to the amount of amino acids that go periphery, i.e. the exposure of skeletal muscle to the amino acids, because that's what matters. And by the way, again, if you go back to Johnny Biolo's earlier work, we didn't even ingest it. We infused it. We infused amino acids. We used the clinical form called Travosol at the time that we used in patients. So you see what I'm saying? The point is there. It's like all you do is expose the muscle to the adequate amino acids and things happen. Okay. Now we complicated it by interjecting the GI in the whole process, right? Which, in all fairness, is how we consume things, right? So that's why I think you have to understand when you start talking about timing and or amount, your first 2 questions, it has a lot to do with those factors.
Yeah, absolutely. I think getting people to understand not all protein is created equal is a tremendous
That's the
tremendous thing.
biggest lie told. That's the biggest lie
Yeah,
told. I say that all the time. I don't care if you're a vegetarian. I have no like emotional
right.
response. I have no cult
Yeah,
feelings about this whatsoever.
yeah,
You do
yeah,
you. I'm just telling you that the data are the data. This is what politics could learn from, right? The data are the data. All right. You
yeah,
lie all you want, but the data are the data. Here's the
yeah.
data. You can lead your life. You can eat what you want. It's up to you. I don't care. But I'm just telling you what the data says. And I just told you the essence of that. It's like, yeah, you can get the amino acids from anywhere, but by the way, calories come with them, right? And the
Right.
digestibility is not as good. So
Yeah.
you decide how you want to play this game.
Yeah, no, absolutely. Absolutely. Well, related to that, we'll get to the last one. Because some people might think a higher protein diet might be unhealthy for, many reasons, kidney health or bone health or cancer or cardiovascular disease. What do the data say related to that?
Yeah, I will point again. Actually, Stu Phillips and colleagues, it's been several years now, 5 years or more ago. Uh, Stu did, actually Joey Antonio did. There's a number of people who actually did studies and or reviews. I think Stu's was a very nice review that discussed exactly that. And the bottom
Hmm.
line is in a healthy person, there's no kidney issues. Now, I mean, as you know, with kidney disease, that's a challenge you have,
Right.
is that they can't excrete the urea and nitrogen or ammonia compounds. So if bad enough, you have to dialyze them, et cetera. And I did work earlier in my career with a guy named Dominic Raj at University of New Mexico Medical School out there, looking at end-stage renal disease patients. And one thing was, to be honest with you, it begged for the essential amino acids. It begged for it. And I'll bet I tried 6 to 8 times to try to get a project funded in that regard and was not successful. Okay. And it just begs for it because of exactly what I mentioned to you. The essential amino acids are very effective. They're non-ureagenic, which is perfect for that population. And that population tends to be sarcopenic because they keep them on
Hmm.
a protein diet. I think, again, it's one of those areas that's lagging behind, in my opinion. It begs for things of this nature.
No, yeah, there's still a thought that consuming higher protein diets are going to affect your kidneys in people
Yeah,
that
they're
don't.
not. You know what? When I got into this like 35 years ago, um, I went to Florida State for a graduate degree, and great people, great program, but they were a lot of them were RDs from the old school, and that was the old
Mhm.
school mentality. It's like too much protein
Right.
is bad, too much protein is
Yeah.
bad. I think now, and it's been 30 years of research for us to show that not only is it not bad, it's preferred, especially with aging
Right.
and some of these other morbidity states and it has again it has all the data shows that it's quite healthy and no problems whatsoever. So
Yeah, and I think when people think about, say, cancer risk or cardiovascular disease risk or things like that, and there might be data that suggests or gives the illusion that consuming a higher protein intake is going to contribute to certain disease risk. That is separate from the amino acid component, right? Versus if you're consuming a bunch of processed red meats, things like that, that is separate from the actual amino acid composition of that protein food. It's this everything else
you're
on top of
exactly
that.
right. That's the first major point. And the second one is you ask 70,000 people about beef intake, but if they're eating a burger, they're eating fries. What do you think the problem is? You think it's the burger, or you think it's the bun and the fries that go along
Right.
with it, and the ketchup
Right.
that has sugar in it too, by the way. All right.
Yeah.
So I think people, and by the way, this is political, and it's very common to the things that people need to understand about politics. You just need to drill down and like find out what the data is, because most of what you hear is bullshit. It's just that way. It's that way in
Mhm.
every field. Okay. So let me recap for your audience here. We did talk timing and amount, which again I think they're related, and take the reductionist approach of the amount of essential amino acids and how quickly you can get them out there, and how much you can get them to rise. And then I think the amount of protein in the diet, becomes self-limiting too. By the way, to Joey Antonio, I don't know if you remember this. Several years ago, Joey did this study at Nova.
Yeah.
We gave him like 4 grams per
4.4.
Yeah.
I show that study to my classes.
Yeah.
Like 4.4 grams per kilo. Yeah.
Which was ridiculous, right? Nobody, I mean, even the guys who are the world's strongest man, there's probably eating 2 point something. But his point was, all right, we pushed the excess, we did all the blood measures, nothing. So the point is that this whole myth about acute protein intake and all this, and by the way, it was self-limiting, right? They were always complaining that they couldn't eat that much protein. Right. They had a lot of GI distension, gas, etc. So I mean, again, biology is very conservational. It's very limited in that regard. So I mean, that was certainly an upper bound he tried to push, but the take-home message was still there, right? It's like no harm, no foul. Hard to eat.
I showed that study, and then did a follow-up study at 3.4. And in Colombia, I asked them, hey, the first study was 4.4, I showed my students that. And then the subsequent study was 3.4. Why did you change it? Why did you lower it? And he said, it was so difficult to get people to eat that much protein.
Yeah, they couldn't eat it. Yeah, exactly. They couldn't eat it. Yeah, and they were getting it free too. So what's that tell you? It was college students getting it free. Yeah, no, that's exactly right. So that's why I said it somewhat becomes limited, and nobody's ever been able to. And why would you test this, and who would pay for it? Nobody's ever been able to test like 3.5 grams per kilogram, for a year and see what that does to your kid. Again, it doesn't make sense. Doesn't make sense.
Yeah.
So again, I just think it's a myth that's perpetuated through the old registered dietitian of 40 years ago plus. And, that's the thing with these myths and these, urban legends, if you will. Once that train leaves the station, it's hard to slow down.
This was awesome. Appreciate your time and your knowledge. And it's always good to talk with you. So
my pleasure, Kurt, my pleasure. If you need anything, don't hesitate to holler. Okay.
Okay. We'll do.
Am I going to say what I said?
I'm not sure yet. I think so, but I haven't got that definitive yet. I'm hoping to.
I hope so. I hope so. Please try to come and bring Sam with you. It'd be great to see you both again. So we haven't broken bread since Medellin 2 years ago there, right?
Yeah. That was a good time though.
It was. Let's do it.
Yeah.
All right, buddy. Thanks a lot. I appreciate it.
All right, Arny. Thank you. Appreciate it.
Yeah.
All right. Take care.